Continuing on with the TMCC Library Open Genealogy Lab Outstanding Guest Speaker Series, today we would like to welcome Dick Tolman. Dick is a twice retired medical scientist with a PhD in biomedical chemistry with a DNA chemistry specialty, who has remade his career as a pro bono genealogical research and writer and volunteer at the Nevada Historical Society. A previous winner of the National Genealogical Society Best Family History in 2006, he has published source genealogical essays on troubled subjects on his website, which is www.29deadpeople.com, and as well as in the genealogical literature. His lecture for today will cover DNA and genealogical research, complete with examples of successful efforts. So without further ado, I would like to turn the helm over to Dick Tolman.
Thank you, Sue. Studies in DNA, family lies are the best kind. The reason that they're the best kind is because they're most likely to stimulate you to do research and find out the truth. This is actually how I got started with genealogy more than 50 years ago. Yep, I'm that old. At that time, I was a young man. I was interested in my genealogy, and there was one big hole that I couldn't deal with. And when I talked with relatives, older relatives who might know something about it, they either didn't know anything or they wouldn't talk to me about it. They just said, this is a shipwrecked family. And so this was very provoking at the time. And I spent, even though I had an active career, I spent spare time and vacations trying to figure out all about this family. I eventually did solve most of the problems and publish the research. Actually, I won the National Genealogical Society competition on the best family history in 2006, and the history was published in the HistGen periodical, the Register. So it is important. These lies are not malicious. Mostly, they're just self-protective. People don't like to talk about things that are unpleasant and uncomfortable. This can be any variety of things. So a lot of times, that's really their solution, is just not to talk about it. And then also, everyone wants to be related to somebody famous. So a lot of times, this wish ends up in their family tree. I remember my aunt told me we have a Davis line, and my aunt told me we were related to Jefferson Davis, well, which turned out to be not true. Relatives have actually disclosed their genealogy on these ancestry family trees and other places. They put down all they know and all they can prove. And then they add just one more generation, a best guess. Unfortunately, others copy this and accept it as the documented truth. So I am personally convinced that this is the source of many of the difficulties that we have with published trees. Certainly, it's the biggest trouble with ancestry trees because they are filled with best guesses that have no support in the record. So it actually, genealogy is all about sources. It's easy to come up with a genealogy if you just can make it up and choose the best guess. But for a person who's really serious about genealogy, we need sources. Now, to remind you, your birth certificate is a primary source for birth. That is, it was written. This record was generated at the time the event happened or close to when it happened. A death certificate then is a primary source for death, but it's a secondary source for birth because the person you'd like to ask is dead. So you're not going to get the best source there. And a book is a tertiary source if no other source is given in the book. The very best proof for any lineage is DNA because that's unalterable. It tells the truth whether you like it or not. And if you're dealing with doing genealogy by DNA, let me warn you that you should be very careful what you promised other people. I've been in the situation where I've tried to help somebody else with their DNA and made suggestions that, you know, I would tell them what was going on, and then later I found out you can be in the situation where you have to explain to an uncle why his sister is not really his sister. So you can get yourself into some real trouble here by promising to deliver. We have something in genealogy called the dog sled rule. If you're not the lead dog, the view never changes. Unfortunately, this is true. And if you're going to do research, you need to become the lead dog. You have to find out what is known and what can be supported with sources. And then you need to do an exhaustive search for historical or paper sources. And once you've done that, then you're ready to go into the DNA. Now, although 23andMe and MyHeritage have pretty much the same search capabilities, this presentation, for the sake of brevity, will talk only about Ancestry. And also elementary principles of DNA and DNA matches, already discussed by others ad nauseum, will be dealt with only briefly so that we can focus on actual research. Most people who do research just copy the work of others, but we're talking here about doing your own research and finding out what is really true. Now, if you've had your DNA sequenced, you will have between 20,000 and 75,000 or more matches. Now, you can't examine them all, and you must find a way to examine them only once. Otherwise, you'll just get old looking at these things. So you need to do a search that will focus on a particular problem. And you can adjust the terms of your search so that you can regulate how many hits you get. Then you have to look at all these hits and mark them with either with colors or hashtags or just write descriptive notes. Ancestry has, and other sites as well, have a number of ways to do this that they have generated. There is also a very useful Google extension called MedBetter DNA that you can install in Chrome that will allow you to display the complete note field. This way you can write a lengthy note that tells about this particular match and why it's important for you. Now, I have found I have the read-only right to several people's DNA that I use regularly, and usually with read-only, you can't make any notes in the field. So I end up printing my hit list and then making my notes on paper. So you may be forced to do this too if you're looking at someone else's DNA. So let's talk about why DNA solves genealogical problems. Ancestry claims to have about 2 million DNA sequences and at $50 a sequence or more. This is really a lot of money anyway. On the other hand, it isn't very many when you consider how many genealogical lines there are. If you're working on a problem that is 6 generations back, your success will depend on whether a key 6th cousin has had their DNA sequence determined. The odds are not good unless you're Mormon or you belong to some other group that is more likely to have their DNA sequence. The good news is, whereas you may have only 100 second cousins, you may have 1,000 or more than 1,000 sixth cousins. So the challenge is to find a DNA sequence that is going to be useful for you, and that has a paper pedigree that will allow you to connect it to the common ancestor or your own DNA. So let's just review for a few minutes a little bit more in depth about DNA. Everyone knows that it's a double helix. Everyone knows that it's a double helix. The information is coded using four bases: adenine, guanine, cytosine, and thymine. And the errors are avoided by having two complementary strands in the helix. A pairs with T, G pairs with C, and these are called base pairs. And if you look at the picture of the double helix over there on the right, there are two grooves. There's a major groove and a minor groove. And they have DNA repair machines that roll down the minor groove and look for aberrations. So if one of your bases becomes mutated by being hit with ultraviolet light or some other chemical moiety, which often happens, the repair machinery will discover this little place where it's irregular, will chop that residue out and put in the right one. So it's a way of mending your DNA so that you always have the correct sequence. Now, DNA is big, 3.2 billion base pairs, and it contains 20,000 genes. I recommend that you watch Ken Burns' The Gene on PBS. If you haven't seen it already, it is really excellent, and it will give you a lot of basic background on what we're talking about here. Another thing that's important to understand is that AT, adenine or adenosine connected to thymidine, this dinucleotide is not the same as TA, because each unit has a front end and a back end. So for a dinucleotide, which is a tumor, there are 16 possibilities, or 4 to the second power. Or for a 3-mer, it would be 4 to the third power, which is 64. So you consider the following. You have a 16-mer DNA fragment. This is one possibility among 4 to the 16th power, which is over 4 billion. Now, the U.S. population in 2019 was 330 million, so it's 10 times the population of the U.S. So you can imagine if you have a DNA fragment which is 100 or 1,000 base pairs, this is going to be unique in the history of the world. So if you're told that there's another person living out there who has 100 or 1,000 base pairs that are identical to yours, this cannot be an accident. This has to be a genetic connection. So now you know that when you have your DNA sequenced, they don't sequence the whole DNA. They only sequence autosomal test sites, which are bigger than 1,000 base pairs, and there are like 700 of these that they routinely sequence. It's not worthwhile to sequence all of your DNA because there are the coding regions. Those are all going to be the same from one to another. If they are not the same, then you'll have a genetic disease, or you'll have some other problem, or you'll be dead. So more about centimorgans. They talk about centimorgans as being a measure of length. They are not a measure of length. They are a measure of gene function. That is, a 1 centimorgan piece of highly functional DNA can be short. A 1 centimorgan piece of more structural DNA can be very long. So it is a safe assumption that a centimorgan is about a million base pairs long. And since DNA comes in pairs, over here on the right is shown a pair of chromosomes. They are bound to one another around the middle, and this middle place where they are bound is called the centromere. And so if you have out on the legs of the chromosome, there you have more likely changes, or when you inherit sequences from your mother or your father, you're more likely to inherit these end pieces of the chromosome, and those areas next to the centromere are more difficult to share. And this explains why you can have DNA, pieces of DNA, which may be 400 or 500 years old, that you've inherited intact because they are close to the centromere. So just giving you some perspective there. So let's keep going here. So you have 23 pairs of chromosomes, 22 autosomes, and 1 sex chromosome. And of course, Y-DNA is concerned with the Y, with one of the chromosomes of the sex chromosome. But for our purposes here, we're talking about autosomal DNA. That's what you have if you have your DNA sequenced. So only 10% of your DNA specifies the sequence of proteins actually used in your body. So all the enzymes, all the other machinery that you use to run your body, that's all specified in 10% of your DNA. And those are called the coding regions. And they're going to be either conserved, they're the same. They're the same for us. Even a chimpanzee has very similar coding regions that's only different in a few areas. And, you know, they now have the sequence of the Neanderthal man. They've shown that the Neanderthal man has virtually the same coding regions as us, with a few changes that allow it to be identified if you have inherited this kind of DNA. The other DNA, the other 90%, is useful for other reasons that it remains still obscure to us. We're still discovering what some of this other DNA is for. Some of it is structural, depends on, and it controls the folding of the DNA in your nucleus. Or it may just be genetic baggage that you have inherited from your ancestors. A lot of times there are repetitive sequences in this 90%, and these are repeated time and time again. And it may be one of the things that is absolutely defining about you, that you have 47 copies of this repeating DNA, and other people may have 46 or 50 or something like that. So there can be subtle changes, but they all mean something. So the autosomal DNA test sites that I've talked about are not from the coding regions, but are from other highly variable regions. So they've chosen the areas that are most variable, and that is what you see, and that's how your matches come from these areas. So autosomal DNA is useful for identifying living relatives by determining the amount of shared DNA, or for identifying a common ancestor. Now, I should say something at this point about solving genealogical problems. In this seminar, I'm talking about solving problems going back 100, 200, 300, 400 years. But you may have other reasons for doing genealogy research. Perhaps you're adopted and you're interested in identifying your birth parents, or you have some tricky stuff going on and you're interested in figuring out who your cousins are. I'm not going to be talking about that, but that's a special problem, and it requires a special strategy for dealing with your DNA. So that's a subject for another lecture. DNA, or autosomal DNA, is advertised by Ancestry as only being reliable for 6 generations back, roughly 6 generations. Used to be 4 generations, now they've changed it to 6. This this is an untruth. The reason they say this is because the shorter the sequence of the match, the more computing effort it requires to identify it. So if you've got a second cousin and you share a lot of DNA, 300 centimorgans or something like that, that's cake. The computer can find that in a hurry, and once they identify that you got 10 bases in a row, they can go back and sequence all 300 centimorgans, no problem. But if you've only got 5 centimorgans that you're sharing with another person, this takes a lot of computing power, and they don't have enough computing power to do this for all their customers. So they tell you that they can only go back 6 generations. But actually, they can go back a lot further. And if you look at DNA, if you look at Ancestry, if you look at Ancestry 2 months ago, you got different hits than you got today because the computer's been working 24/7 since the last time you logged in. So now you'll have more hits. So you should continue to go back to Ancestry and do the same search over again, and then look at the bottom of the search where you have the low centimorgan hits, and you'll find you have a whole bunch of new hits because they down to 4 and 3 centimorgans. Those can still be valuable hits because they're still far beyond. They're still long enough to be unique in the world. So don't ignore those low centimorgan hits. And we'll talk about how to go back. But I've already identified several common ancestors that are 11 generations back or more. And so it is possible to do a lot more. As the computing power and time goes by, you'll get more and more useful hits. Okay, well, I have been doing this for a number of years, and I've been able to solve a lot of problems that the family genealogists have worked on for years. I had a distant aunt who worked many years at the Family History Library in Salt Lake City, and she solved problems. She wrote several monographs about the work she had done. But one of the problems she worked on was this Murkins problem, which I'll be sharing with you in a few minutes as one example. But I wish she were still alive. I think she died in the 1960s sometime. I wish she were still alive because she would be thrilled to know the stuff that we have been able to uncover that she worked on most of her life. So, and in addition to these problems here, very recently we discovered an adopted son in one of our lines. He was born in 1740. We discovered that through DNA that he was adopted, and we also were able to identify his birth parents, which is, I think, truly remarkable. So these problems and their solutions are published on my website. I publish essays that'll have all of the details of these investigations, and that's on 29deadpeople.com. It's a free site, and you're welcome to take a look. So this is the first one. This is the first example on the previous slide, the Murkins common ancestor. We had taken the line here on the left, which is my maternal. Raymond is my maternal uncle. Back to the individual here who was in colonial Pennsylvania in 1780 or so, and we knew he came from England, but we just had no idea where he came from. And various people interested in genealogy in the family had tried to discover where he came from in England, but England is a big place, and we were not able to do it. I worked on it in my youth for several sessions and was not able to find his ancestry. But Raymond, we discovered, had a match with Caroline Carter, who lives in Somerset today. She's a grandmother. She lives on a pig farm in Somerset that is owned by her daughter and her husband. And she has done her genealogy back about 4 or 5 generations, and all of her people came from Suffolk. Well, that's what we needed to know. That was the key discovery. So once we knew that Caroline's people came from Suffolk, we went back and looked for Raymond's ancestors in Suffolk and found them in a hurry. And so we filled in the blanks here on the left, using the parish registers from Suffolk, and using other records. I was able to take Caroline's genealogy back to the common ancestor Murkins, who was born about 1700. So it was then a combination of the pedigrees, which we already had, plus some paper research to do the connection, but it was the DNA that told us where to look and identified the common ancestor. So this is another interesting thing, and reads on the problem of, what do you do? What's the first thing that you should do in doing your DNA genealogy? It's important to know that genetic inheritance is random and it's unpredictable. Here on the right are shown Raymond and Carolyn again, 7 centimorgan match. And Raymond got his Murkins DNA from his mother, who's my grandmother. And Verna is my mother, and the left-hand X is me. The right-hand X is my brother. Neither one of us have a match with Caroline because we didn't get enough Murkins DNA. But Raymond had another brother, Arlen. He got a big endowment of the Murkins DNA. Marlon has 2 daughters that have both had their DNA done. One daughter also has no match with Caroline, but the other daughter, strangely, has a 9 centimorgan match with Caroline. So it just goes to prove that you need to have more than one source for your DNA, and older people are more valuable. So a few things to remember first. DNA can only be inherited from the individuals who are on the direct line. Once you get doing DNA, it's easy to get confused, and you need to remember that you can only inherit on the direct line. You can't inherit from a collateral line. And the absence of evidence is evidence, but not proof of absence. So just because it isn't there is not proof that it isn't there. It may be that the right person has not had their DNA sequenced. So the third rule, the frequency of shared matches decreases going back generations. I spoke a little while ago about solving problems closer to home, about your birth parents and other strange connections that can exist. For those kinds of studies, the shared matches is very important. In fact, it's the key to solving those problems. But for going back 100, 200, 300 years, the further you go back, the fewer shared matches there are. So for most of the things that I'm talking about here, there are no shared matches. There are too many possibilities, and you're too far removed from current events. There may be many matches that contain pieces of ancient DNA, but these are only useful if you have a pedigree connection to the ancient line. And this is very important. So when you do your narrowing down search and you get all of these matches, what you're looking for is a match that has a significant pedigree attached to it. and we'll talk about that more in a couple of minutes. So you need to get reading rights to as many pieces of DNA as you can. Now, I have the reading right to my brother's DNA, which has turned out to be very important because he has had several matches that I don't have. So if you can get a sib to have his DNA tested or sequenced, that's a good move. But having an older person having their DNA sequenced, that's even more important. Because every time you go back a generation, you're going to double the amount of ancestor DNA. So that's the key. If there are older people, aunts, uncles, a grandparent that's still alive, pay for their DNA to be sequenced. It's going to be worth it. Now, the average centimorgans for a fourth or a fifth cousin is about 25 to 35 centimorgans. So you need to be starting, and there will be several pieces of DNA that will be outside this range. So you want to start about 50 centimorgans and then work down. So choosing of the right DNA is very important. So let's go now to the other problem I wanted to show you today. I have an ancestor, Nancy Graham, who was born in 1797 in the Western Reserve of Connecticut, died roughly 50 years later in Illinois. There is a primary source for her marriage in 1816 at Madison, Lake County, Ohio, but that's all. So there is a pedigree on Family Search going back 6 generations from Nancy to John Graham of Glasgow. This doesn't mean much. Graham is a fairly common name in colonial America. There were 27 Grahams that fought in the Revolutionary War. So just saying that you go back to John Graham of Glasgow without any proof does not impress anyone, because there's no source beyond Nancy to connect the generations. So our strategy then is to confirm or not to confirm these linkages. And where do you start? We'll start with Nancy's proposed parents, which on Family Search are Samuel and Nancy Sanders or Stevens Graham. And so then I'll want to check autosomal DNA matches beginning about 30 centimorgans for possible descendants of Nancy's parents or grandparents. So this is my big DNA resource. My paternal aunt, she died in 2017, but had her daughter was heads up enough to get her DNA done before she died. And so this shows her lineage back to Nancy Graham. So Nancy Graham is over here in the lower right. And he goes through the Loveland line. So there are going to be a lot of Loveland matches. But I have only shown Nancy's parents as Graham because that's all we know at this point. But after having looked at all of these DNA matches, we know that, or we can assume that that Enid Tolman Seeley has the largest known source of Graham DNA. I have not seen anyone else who has any bigger piece of the Graham DNA. So I'm indebted to Diane Southard here for these categories, which I quite like. The bottom 3 is hers. The top one is mine. These matches that don't have a tree or have a private tree, there's just simply nothing you can do with them, so you just have to draw a line through them. These people are simply losing out. I'm still at a loss to understand why you wouldn't share your DNA, because there's absolutely no risk to doing this. But a tree with up to 50 individuals, Diane calls dating, but it takes a lot of work to find a common ancestor if you only have a pedigree of 50 individuals to start with. Then going steady, that's where a tree that has more than 50 individuals, requires some work to fill in the tree. And if you choose to put energy into this, the geographic location of the tree, of the individual in the tree, has to be encouraging. So if you're looking at Farringtons and you're looking in Flushing, New York, if you have Farringtons in Massachusetts, where the Farringtons first immigrated, that's not a good choice, because you'll have to make the bridge between New York and Massachusetts. So you want to focus just on the ones in New York. So the geographic location is one of the determining things to decide whether you work on this tree or not. And then a married match is one where the common ancestor is almost obvious, but even then, you're going to be required to do some sleuthing in order to make the connection. So in this particular case study, my aunt had roughly 75,000 matches, and I searched for Graham, and it took it down to 260 matches, which is a lot. When you print that out, 10 per page, it comes out to 26 pages, which is a lot of work. And it starts about 50 centimorgans, and took it down to about 8. That's all that was available at the time. Since then, they have added some more matches down to about 5 or maybe 4. But the breakdown of the matches that I went through one at a time is shown in the box here. So the ones that had a Loveland common ancestor were 84. People with no trees were 29. Dating matches 133, so it's very common. A lot of people know a little bit about their ancestry, but not a lot. And then the two bottom ones are the ones that are going to be useful for us. The going steady matches, which are the ones that are going to require significant work, and the married matches, which are also going to require a little bit of work. So this is the result of all of that work. We identified Nancy's parents, which are not the ones in Family Search, but a different set of Grahams, and we found other errors as well. But there were 9 DNA matches to a common ancestor, Samuel Graham, born 1771. This is a different Graham than was identified in Family Search, and we were able to, with paper sleuthing and regular genealogical research, to connect all of these people to Samuel Graham. It's interesting that the most important match here we didn't get from my paternal aunt, but it's a match that both my brother and I had. So we got it from our father, who was the brother of my paternal aunt. So it just so shows that you can't ignore the other pieces of DNA. So this was important for this particular connection, to make this particular connection. So we connected Nancy Graham to her parents through these 9 matches, and then we were able to take the Graham line back another 3 generations to Andrew Graham, born 1658 in Glasgow. All of the Grahams come from Glasgow, but there were several immigrating Grahams, and there was particularly another Graham, Hugh Graham, who immigrated from Glasgow, but his family spent one generation in Northern Ireland, which was typical at the time before they emigrated to America. And he settled in New Hampshire, and his children married into the Coltrane family, which turns out to be one of the other families of interest because Samuel Graham married Hannah Coltrane and connected to the Coltrane line. So not only were we able to follow the Graham line back to the immigrating couple, but we followed the maiden name lines back and took them all back to their English origins as well. And that was the Hun line, and we had 9 DNA matches that read onto that problem, either with the common ancestor Samuel Graham or Samuel Hun, or his father, or Nathaniel Hun, which is one generation removed from the immigrating ancestor who came to Boston in the early 1600s. We also were able to follow the Coltranes back to England, and also the Maxim line back to England. The Maxims immigrated to Plymouth, and most of them stayed in Plymouth, but there was one family that went to New York, and it was that family that married into the Coltranes, and we were not able to connect the Rhode Island or Massachusetts ones with the New York ones, but there were several DNA matches that showed that this was the correct line. Okay, there are also the negative results, which turn out to be important because they are supportive. I also looked for Sanders, Sanderson, Stevens, the proposed parents of Nancy Graham, but I was able to find no matches that supported this. And the John Graham, the son of Hugh Graham, who came from Glasgow but went to New Hampshire. His wife's name was Wallace. I also searched for Wallace. Was not able to find any Wallace connection either. So, in summary, a combination of paper resources and DNA matches proved the Graham pedigree to Andrew Graham, born 1658 of Grahamton. Apparently, in 1658, Glasgow was not a very important place, but there was only a collection of houses that they called Grahamtown, and this is on the current site of the Caledonian Central Railway station. So none of it exists now, but all of these Grahams lived in this little village. We found several errors, actually 5 in number, in the connectivity linkage of the published pedigree. And we found new family connections to the pedigree that no one knew about beforehand. And also we found that some proposed family connections were disproven, namely the connections. There were a number of proposed connections to the New Hampshire Grahams, and those were then disproven.
Okay, let's see here. Let me open up the chat box here. Okay, not so far. I'll go ahead and ask the class to go ahead. You know what? It might actually be better to let people with questions to turn on their microphone, not their camera, but their microphone. So I'll let the class, if you have a question, go ahead and please unmute your microphone and go ahead and feel free to ask the question directly to our guest speaker. And while they're thinking about questions, let me go ahead. I had a couple. What is your feeling about GEDmatch? I know you concentrated specifically on Ancestry.com, but of course Ancestry.com has an emphasis of more tests in America than overseas, whereas MyHeritage, has more tests overseas than in America. So, what is your feeling about GEDmatch?
I don't use it. I went to Roots Tech this last February, and this I'm looking for them to have changed their sharing policy, but GEDmatch is controlled financially or supported financially by several medical groups that are going to use this DNA to do searches for aberrant DNA. And I prefer not to have my DNA searched in that way. I'd like to have a little bit more control over it. So if you upload your DNA to GEDmatch, and this is also important for Ancestry. In Ancestry, you can go into settings and you can change it to do not share. You don't have this option in GEDmatch, as far as I know. I'm looking for them to change this in a short time, and then I'll be glad to use GEDmatch. GEDmatch doesn't bring anything to the table except to locate your DNA on a particular chromosome, which would be nice, but it's not essential for doing genealogy. At least in a few, it is going to be useful in a couple of situations, but for most situations, it's not doesn't bring anything to the table.
Let's see here. You talked about the Ken Burns video called "The Gene." We have a lot of Ken Burns videos in our streaming video collection. But I went in while you were talking and I looked and I couldn't find one called "The Gene." Is that the correct name of the one you're talking about?
It's called "The Gene." We streamed it from the PBS website, I would say a month ago. So it is new. They showed it on PBS on the local station a couple of months ago, but we missed that. But we were able to stream it. So if you have the ability to stream from PBS, you can still stream it today. Just go to search and search either Ken Burns or "The Gene."
Okay, I'll do that for the classes. And if I can find it, I'll include it in the links I send out next week. Let's see here. Now, I'm curious, how did you get the name of your website, 29 Dead People?
It's just chosen at random. We lived for 25 years in New Jersey, and our house number was 29. It's a prime number, so I like it. And so when we needed to start a website, we needed to start with something, so we chose that.
Okay, great. That's a great story. Thank you. Let's see here. In our class, we've talked a lot in the past, and one of the most common questions, obviously for financial reasons, one of the most common questions is, do I really need to test my brother or my sister if I've already tested?
The answer is yes.
Yeah, you know what? I was just going to ask you that because we've had other guest speakers that say no, but I can really see the value of testing.
They're wrong. Sorry. I hate to be blunt, but there's no other answer. It's the randomness will get you. And these, as you can see with this work that we've done and on the other problems that we have worked on, it's many times a single match that makes the difference. You think with all that many matches that you'd have more than one. Well, it doesn't happen that way. Sometimes it's one match that makes the difference. And you can't predict. Now you think, well, my paternal aunt is going to have all the good matches because she's got more DNA. That doesn't mean anything, because you can still, if you got it from a different line, we got it from our father. My brother and I got it from our father, who got a different portion of the DNA. So we have matches that she does not have. So it's important to get other people and to be able to have read-only access to their DNA.
Actually, that's been my experience as well. I've got a brick wall on a Harris line in my own family tree, and I got to tell you, I have been really scouring all the different. I've got my brothers tested. I've got my half brothers tested. I've got my uncle tested. I've got two of my second cousins tested, and I got to tell you, not all of them are showing this Harris line. And some are. So I'm going to take the example you gave today about going steady and that kind of thing. I think those are really great suggestions on how to start narrowing down that Harris line.
Yeah. Well, we owe Diane Southard for that. I thought that was a very clever way to break them down because they need an identifier like that. But, it's important for another reason. My father died at age 38. I was the oldest of 6, and I was only 14. So I'm one of the few who still remember him. But because of that, and because we lived far away from the other relatives, none of my sibs or myself ever got to know my father's side of the family because he died, and without him to introduce us to these people, we just didn't meet him. So I've had this handicap all of my life that my father's family has been virtually unknown to me. So when I did this DNA stuff, I realized that I sure needed to have some more relevant DNA. And unfortunately, although there were 12 in my father's family, they were all dead, except 2. And I couldn't find one of them. And the other one, I knew where she was, but we'd driven up that way through Oregon. I should have stopped and introduced myself, but I didn't. It's too hard to go cold to somebody's doorstep and say, I'm your relative. So she died 2017, and I decided I would build up the courage to talk to my cousin, her daughter, that I knew from what she had published that she was interested in DNA and genealogy. So I built up the courage to email her, and I got a very nice response. We met for the first time this last February, and she was generous enough to share her mother's DNA read-only access with me. Made all the difference in the world to have another older relative on my father's side of the family. Plus, now I get to know my cousin, who is really a charming person.
I've actually had similar situations, but I've actually had the nerve to go up and knock on a stranger's door and introduce myself. My husband died 1,000 deaths. He waited in the car and told me not to do it, but I did it.
Good for you. Good for you. I wish I had that kind of courage.
Okay, one of my students emailed me and said that the name of the PBS show is called The Gene in Intimate History. It's a 2-part documentary on PBS.
Yep.
And then Jill, one of our students, would like to know, could you go back to that slide and share it again about dating, going steady, and getting married?
Okay, your wish is my command.
And I'll tell you, that makes such good sense, because the numbers are overwhelming when you start looking at your DNA hits.
Yeah, well, you can find out that if you just add a place, you think, well, 260 is too many to look at, so you want to add another restriction to it. Any restriction you add gives you no hits. So it's the way the ancestry algorithm is constructed that they look for an exact match. Well, a lot of times you'll have a match, but it isn't exactly what you searched for. So you have to be very lucky or very good to restrict them anymore. So I'm happy with 260. I don't mind looking at that many.
And then how did you just further sort it after you got your 260? How did you further sort it? Did you sort it by centimorgans? Is that how you did it?
It's automatically searched by centimorgans. So the first hit is going to be like 50 centimorgans, and then it'll go down to 3 or 4 or whatever their computer is able to calculate right now. So it's already organized by centimorgans. I print them out because most of my DNA, the DNAs that I look at, are read-only only. So I can't write in the notes field. I can't do any of those things. So I just print them out, and they print 10 per page. And then I go through the list, and I make notes on the paper, which ones are good and which ones I put the oldest hit on in their pedigree. and then I can identify them. So that I go through the 26 pages, and then I'll have a few of them that are highlighted, a couple of dozen perhaps that have useful lineages, either a partial pedigree that is obvious that makes them relatives, or else something else that makes them useful.
So you're simply going to the DNA tab in Ancestry, going down to the matches, and then when the matches open up, you go to, on the right-hand side, upper right-hand side, you go to the search box, which brings up the 3 boxes: match name, surname and match trees, and birth location, correct?
Right. And then right-click with your mouse, and one of the options is print, and it'll print your all those pages, 10 per page.
Understood. Okay. Now, just out of curiosity, do you also include similar names, since that's an option underneath that search?
It depends on the name. A lot of times you have to truncate the name and use a wild card. Oftentimes, I did a search for Farrington not long ago, and it gave me Farrell and Farr as similar, which trebled or 10 times the number of hits. So I find that the algorithm is not strict enough for Ancestry. So I can't choose, usually I can't choose common names. If you had something like Wisniewski, probably you could do that, but not with something like Graham.
Understood. Okay. Now, let's see here. One of the students would like to know if you do other webinars or if you have any YouTube videos.
This is my first webinar. I've given other lectures, but this is the first one on the computer. I certainly have lots of material for doing other ones, but this is simply one of the things I've been working on most recently.
And then another student just chatted and said, this is wonderful that she's learned this, because she just sent in her DNA kit yesterday. However, she sent in an mtDNA test. And now, because we do have some beginners and first-time people here in the class today, would you mind, I know this is a little bit off the topic of autosomal, but could you talk about briefly the difference between autosomal, MT, and Y, just for the benefit of the new people?
Of course. Mitochondrial DNA is a maternal, it follows the maternal line, because you have in the center of the cell, you have your nucleus, and then in order to provide energy for the cell, you have these little floating organelles called mitochondria. And since they're the ones that respire, they're the ones that take oxygen and convert it to water, and do all the other work, the energy work of the cell. They need to be able to work really fast, so they can't send messengers back to the nucleus to get instructions. So they have their own little piece of DNA in the mitochondria, and that's called mtDNA, or mitochondrial DNA. It's inherited exclusively from the mother. So if you go through what's called the umbilical line, which is all the females in your pedigree, so mother, grandmother, great-grandmother, great-great-grandmother, you will have inherited your great-great-grandmother's mitochondrial DNA, because that's going directly from mother to daughter down to yourself. And of course the men get it too, but it's always from the mother. So the problem with mitochondrial DNA is it's small. It's only 12,000 base pairs, and in order to have a match, it needs to be nearly identical to other matches in order for it to have any meaning. So you need to have nearly a perfect match in order to prove the lineage. So I have not worked with mitochondrial DNA, but there's a wonderful book that tells all about this. And I've forgotten the author, but the name is called The 7 Daughters of Eve, which follows mitochondrial DNA back to the beginnings. It's a fascinating book. I couldn't put it down.
Actually, our library has that book, and it's by Brian Sykes.
That's the one. And I did, okay, I can also talk about Y-DNA. I have done a Y-DNA study. A Y-DNA is if you have it done or you're going to do any research with it, you should go to Family Tree DNA, which is run by what's his name? Greenberg is the owner. It's actually from Israel. They are very good, and they have a surname file for all the people. Now these, just like mitochondrial DNA is for the umbilical line just on the maternal line, because Y-DNA, the only people that have Y-DNA are men, because in the sex chromosomes you either have X or Y. The women have two X chromosomes. Men have a Y chromosome and an X chromosome, so they get the the X from their mother and the Y from their father, and the Y encodes all of the proteins that are important for becoming a male. They have something called um, they have when a fetus is developing in the womb, it's a female until about the fourth month, when if the Y chromosome is present, it is transcribed at that time, and then the female genitalia are all changed to male. And this is called Wolffian differentiation. So this Y chromosome is important because you get this only from your father, and so it follows the patriarchal line, your surname line, and it goes back reliably much further than you can without autosomal DNA. So I was interested in a gentleman who immigrated and got property uh, from in Pennsylvania in Bucks County, and we recently followed him. But I was interested in following the Y-DNA, so I was able through the Y-DNA file and recruiting other people to have their Y-DNA determined on Family Tree DNA. I was able to reconstruct all of the descendants of this Thomas Terry of Bucks, born about 1650.
Wow.
Yeah, and that's published in Crossroads, which is the organ of the Utah Genealogical Society. It's also on my website. And I'm getting ready to make an update to that, because some of the things that were kind of tentative in the end of that essay, I've now proven with autosomal DNA.
Wow. Well, speaking of autosomal DNA, Beth would also like to know, since she just sent in her MT test, she would like to know, do you also recommend that she take the autosomal test as well?
Absolutely. And you can have it done anywhere, but you should, whenever it's done, you should upload it to Ancestry. They're the ones that have the most useful algorithm for sorting.
Wonderful. Okay, let's see here. Anybody else have any questions? Feel free to unmute your mic and ask the question if you do.
Sue,
Yes?
this is Marlene.
Hi, Marlene.
I have a comment. I have managed DNA projects at Family Tree DNA for 15 years. And first, I'd like to say the company is not an Israeli company. It's owned by two men who live in Houston, Texas, and are .com millionaires. And that's how they got the money to start up Family Tree DNA. Because when they sold their .com millionaire company, they retired and did genealogy, and then realized that they had DNA questions, and they started the company. Anyway, a couple of comments. Mitochondrial DNA for most women in the United States whose maternal line comes from Western Europe can be very disappointing, because most of those women are in the H haplogroup, which covers millions of people. So, and it is also, as Dick said, it's also limited in what you can find out with it. I always recommend my female subscribers do the autosomal DNA test. And if their pockets are deep enough, then I suggest they do the mitochondrial test. And for all my male subscribers, I recommend first that they do the Y-DNA test. Many drag their feet because it's more expensive. But quite frankly, it doesn't do you any good to do a lot of research on a specific surname if your DNA is some other surname. So I always recommend the Y-DNA test, and then I recommend the autosomal DNA test, because it deals with the women who married into a particular family really more often than it does with the male line of descent. So that's my comment.
Thank you. Let me get, before we get information, comments from our guest speaker, let me kind of chime in. I think My Heritage is the company that's located in Israel. They were actually our guest speaker a couple weeks ago. If everybody who's in class today got a chance to catch that, that was a really great presentation. Anyhow, speaking of MT.
The reason I thought they were from Israel, and I knew that the owner was from Texas, because I've talked with him a couple of times. He's a great speaker. Is that he is Jewish, and
Yeah, he is.
they have a great collection of Jewish lines. And if you look at Y-DNA, the Jewish lines are the most confusing and the most difficult to follow, which I guess is perfect, because he's got something to work on and to be a challenge to him. When we lived in New Jersey, I had a neighbor who was Jewish, and she asked me to look at a particular Jewish line, and I've done a lot with this. I've joined the Jewish Genealogical Society, and I've done a lot of work back to Spain and using Southern Germany, because I read German well. So anyway, I've looked through all these German lines, and all of this stuff is all on the family tree DNA.
I personally, Dick, I love family tree DNA. I've done a lot of testing through there on not only myself and my immediate siblings, but also on my distant relatives as well. One thing I wanted to comment on, especially on the Y-DNA through family tree DNA, their surname studies have been so beneficial to me, especially for the Y lines, because it, like for instance, one of the common surnames in my tree is Bartlett, and there are so many different lines of Y Bartlett DNA. And one of the things they do in the surname studies is to subdivide those Bartlett names into different family lines of Bartlett. So you don't go chasing a Bartlett that's not in your family line. So I would strongly recommend, especially for those people doing the Y-DNA test, to look into surname studies through family tree DNA. And then also regarding mtDNA, to kind of like talk about what Dick's point was earlier in the conversation, I did a MT test through family tree DNA, and I came up with 0 matches, absolutely none. And yet I had two of my second cousins who descend from the same female line. I had them do the MT test, and they came up with many, many, many matches. So when I approached family tree DNA for a possible explanation, they looked into my kit, and they said that there had been a mutation between my mother and myself. And they went into great lengths of, and I won't, bring that into today's conversation because that's a different topic. But that might be something to consider, when you do an MT test. If you don't come up with some good hits, you may want to consider testing other people on the same MT line.
Along that lines, I'll add one other thing. Back when I was first tested, mtDNA was the only thing available. And I eventually did the full mitochondrial sequence, and that's the only thing I'd recommend because anything less than that won't give you any information. And unlike most American women, although my ancestors came from Western Europe, it turned out that I have a very rare haplogroup. And my female ancestors at one time were apparently members of the Sami tribe in Lapland herding reindeer.
Wow.
And I submitted it to GenBank, and my genome is one of the defining genomes for my haplogroup, which is V3C.
Leon wanted to comment that he doesn't think, and I just double checked that you can import your DNA autosomal test results from another company into Ancestry. It looks like you can take Ancestry and put it into another place like GEDmatch or other locations, but I just Googled it real quickly here, and it does not look like you can take a test unless something has changed from what I just read on the internet and import it into Ancestry. Are you aware of anything different, Dick?
No, I'm not. I didn't know that. I know that MyHeritage will take it from any source.
That is correct. I recall hearing that myself. So, okay,
Family Tree DNA will take it from 23andMe and Ancestry, and I think MyHeritage too.
Yeah, just that looks like the Ancestry will not allow you to do that though. Like I said, I just Googled it, and unless something's changed from what I just read, it does not look like right now you can. So
It used to be different. I guess I'm not really surprised.
okay. Any other questions from the class? That was some good questions. If there's anybody else, you can please feel free to unmute your microphone. Let's see here. Leon, why don't you go ahead and if you can, unmute your microphone, because that question, I'm not quite sure what you're meaning by it. So if you could,
okay,
is it possible?
Can you hear me?
Yes, I can, Leon.
Since we're at the end of the class, I have a more of your line question, okay? It has to do with RNA sensing.
And what is
Did
your
you hear
question,
that?
Leon? Yeah, we heard it, but what is your question specifically?
The coronavirus up till about the middle of April was labeled a negative RNA sense virus. And in my research, this is my hobby. In my research, I could not find any negative sense RNA coronaviruses. Then in about the middle of April, everybody changed and said, no, it's a positive sense RNA coronavirus. Can you possibly give some reason why that was a mistake?
No, I can't. I know that the coronaviruses are a small group of viruses, and that they can be either positive or negative sense RNA. I don't know any more than that, except virtually all of them in the whole class are dangerous in some way because they have the capability of being integrated into a human genome.
That's my understanding too. Zoonosis, I think it's called.
Yes.
Okay, thank you very much. That has bewildered me for a very long time, because how can you make that such a simple a test? Make a mistake on that. I mean, sensing RNA, whether it's positive or negative, is a very easy test for microbiology. How could they make that mistake? I've always been confused about that.
Well, I don't know. I don't think it makes a lot of difference. It's kind of an academic issue, whether it's a positive or a negative sense RNA. They are dangerous. I'm not a virologist, but I have a question out to my local virologist too along this line, wondering if the the virus is able to infect quiescent muscle cells, because I thought it was unusual that all of the outbreaks of coronavirus were in meatpacking plants. And if you say that, if you believe, I don't know whether to believe or not, but if you believe that this came from a Wuhan meat market, well, I wonder what that means. Whether the virus then infected the meat, and the meat people consume the meat. That is possible. The virus can be grown in culture. Not this one, of course. They're doing it right now. But other coronaviruses can be grown in cell culture, and there'll be a list of cells which they can infect, only differentially infect. So the question is whether they could infect meat cells or muscle cells, and this could be a reason why there are so many outbreaks in meatpacking plants. I'm sure the meatpacking plants don't want to hear about this, but otherwise, it's very difficult to explain why the virus is going to be hanging around in a Wuhan meat market unless it's able to infect something which can then infect humans. Certainly, if it comes from bats, bats are not going to be drawn to meat markets, as far as I know.
Well, it's my understanding is that these meat markets are right outside the restaurants that feed them. The meat markets and the restaurants are one entity, and they usually keep the animals live until, it's time to serve them.
Yeah.
So, there's lots of possibilities there.
Very familiar with it. I went to Hong Kong a number of years ago and went to one of these restaurants, and that's exactly the way they work. You went first to the place where you picked out what you wanted to eat, and then you go in the restaurant and they serve it to you.
Yeah. Okay. Thank you very much. Okay. I'm sorry, Sue. I thought we were finished with regular questions, but I had to ask Dr. Tolman that question.
That's fine. Yeah, it's an interesting question that you asked, and kind of timely, actually. Okay, so this is the class's last chance to ask questions. I'll give you a few moments. You can either turn on your microphone or put it in the chat box. We still have 29 people logged on.
I have another RNA question. If
Yes.
Nobody else has any more questions.
Go ahead, Leon.
Okay. Under the 5 nucleotides, okay, including uracil, okay.
Okay, RNA.
RNA. I'm sorry.
4 for DNA, 4 for RNA, but thymine is replaced with uracil.
Correct. Now, during the codon chart that we have, the 64 codon chart, the third nucleotide can be replaced with another nucleotide question mark called inosine.
Yeah, inosine is related to guanine, actually. And it can be. There's also pseudouridine, which is another rare base.
Okay. From an academic point of view, are those considered nucleotides?
Oh, of course. Yeah, they are.
Okay, so there's 6 or 7 of the nucleotides.
Yeah.
Okay, thank you.
It's really beyond the scope of DNA. This is little details that are important, but not important to most people.
Yes, this is from an academic point of view.
Yeah. Well, they're most of them are made by modification of the DNA itself. So where inosine is involved, they have a specific enzyme. This is located in a particular place. The DNA or RNA has a conformation which allows some places to be more available than others, and this is in an available place, and the enzyme comes along and converts guanosine to inosine. That's how it happens.
Thank you very much, Professor.
All part of our friendly service.
Thank you.
Thank you, Leon. That was a good question. Okay, so it doesn't look like there's any more questions. We got a lot of people saying thank you, and they're going to be watching the PBS program that you recommended. And I wanted to say thank you for allowing us to record the video today. And I should be able to get you the video link next week. It'll be a streaming video on the school YouTube channel. And let's see here. Oh, one last question. Do you mind if through your website, if my students reach out to you with individual questions?
Of course not. That's what it's for.
Wonderful. That's always so helpful. We really appreciate that. Okay, so since my class can't clap, I'm going to go ahead and say thank you very much. And I'll let the class know that in order to hang up and exit out of the webinar, just click on that red hang up button. And once again, thank you very much, Dick. It's been wonderful and very informative, and we do deeply appreciate your expertise as well as your time.
My pleasure.